It looks like everybody's joined who's already got admitted into the waiting room, so we'll get started. Today we have Dr. Ahmed El-Sohemy. Sorry, I'm going to mispronounce that. He is a professor and associate chair at the University of Toronto. He earned his PhD in nutritional sciences from the University of Toronto and a postdoctoral fellowship at Harvard. The goal of his research is to understand how genetic differences modify response to diet for optimal health and performance. He has published over 180 peer-reviewed articles and given over 300 invited talks around the world. He is on the editorial board of 10 scientific and medical journals and served as an expert reviewer for more than 30 other journals and 12 granting agencies. He is currently editor-in-chief of the Journal of Genes and Nutrition and specialty chief editor of Nutrigenomix for Frontiers in Nutrition. He has served on Health Canada's Scientific Advisory Board and several international expert advisory panels. He's also received the Centrum Foundation New Scientist Award for Outstanding Research by the Canadian Nutrition Society, the Mark Bieber Professional Award by the American College of Nutrition, and the Stanley Wallach Award by the American Nutrition Association. Welcome.
Great. Thanks very much, Kathy, for the kind introduction. And hello, everyone. I was just telling Kathy that my kids are off school today because of the disruptions, and so I'm hunkered here in my my garage, the only place where I can have some quiet. So it's a pleasure really to have this opportunity to share with you some of the research that we've been doing at the University of Toronto in the field of genetic testing, and also share with you a little bit of how we've translated some of that science through a startup company called Nutrigenomix with an X. So just by way of disclosure, I am the founder and I do hold shares in the company Nutrigenomix, which provides genetic testing for personalized nutrition for healthcare professionals, so not direct to consumer. So the term nutrigenomics, CS, is really a branch of science that deals with the interface between nutrition and genetics, with an ultimate goal of really applying genetic testing to understand why some people respond differently from others to the same foods, beverages, and supplements that they consume. Whoops. So from a research perspective, we can think of really two sides of a coin. On the one hand, we know that diet can affect genes. Nutrients can turn on and turn off certain genes. And the flip side of that is looking at how variations in genes, the sequence variations, alter the way that we respond to diet. And that's really the focus of our research program. Now, before a gene or a gene product can even act on a dietary factor, genes can also influence the foods that we select. And this is an area that we stumbled across just by chance a number of years ago. One of my former PhD students, Karen Enney, who's currently the Director of Clinical Services at Nutrigenomix, she was looking to try to understand why some people respond differently to an oral glucose tolerance test. Some people who consume the same amount of glucose show a steep rise in blood glucose, whereas others show a more gradual rise. And we honed in on a glucose transporter, but it looked like it wasn't really related to how glucose is metabolized. But what we found was, just by chance, those who have a particular version of this gene just consumed more sugar on a daily basis. It impacted their habitual sugar consumption compared to those who had a different version, the threonine version of this gene. And it turns out that this glucose transporter is expressed in regions of the brain that regulate appetite. So it was kind of interesting that this genetic variation, it was the first study of its kind to ever show that a common genetic variation influences an eating habit, and specifically sugar consumption. So we published this in Physiological Genomics. This was back in 2008, and it was the kind of thing that got quite a bit of media attention. I don't know if you ever recall, the front page of the Toronto Star had this headline, craving something sweet, blame it on your DNA. Not exactly the message we wanted to communicate, but nevertheless, it just illustrates this concept that our genes do, to some extent, influence our likes and dislikes of various foods, and ultimately what and how much we choose to consume to some extent. So at the time, a colleague in the Faculty of Dentistry contacted me and said, I just heard about this study, and as you know, sugar consumption is a major risk factor for dental caries. So he wondered if this gene and another gene that we also showed, it's called the TAS1R2, which is a sweet taste receptor that also influences sugar consumption. We wanted to see if these genes can be directly related to cavities and other kinds of dental caries. So we conducted this study where one of the dental students, Tabitha Chang, was blinded to people's genetics, and she conducted these oral examinations. And what we showed was those who have the combined genotypes, the purple bar that's associated with the highest level of sugar consumption, those individuals have a greater number of decayed, missing, or filled teeth. That's what DMFT stands for. And it was also confirmed by X-ray, as well as this international scoring system for measuring dental caries. Now, I just want to point out that these are all individuals in their 20s, so fairly young adults, and already those who have the gene or the genotype that predisposes them to consuming higher amounts of sugar over the course of their life, they have twice the number of dental caries, decayed, missing, or filled teeth. So we went from this kind of serendipitous finding of a gene that is predicting habitual sugar consumption to showing that that can also have direct implications for health, and in this case, oral health. So since we published this back in 2012, other studies from around the world have also replicated this, even in younger individuals. So in even as little as school-age children who have these genetic variants, they also have greater dental caries. It was also replicated in another population, and yet another study showing it in even older adults, the same thing. So just to demonstrate how robust this finding is in different age groups, different ethnicities, different populations around the world. So this, of course, has important implications because oftentimes these people are over-consuming sugar in a kind of a mindless manner. And so if you can become aware that you have this genetic variant or that your children have this genetic variant, then perhaps you could be extra mindful of ensuring that A, they're practicing good oral hygiene, but more importantly, that they're also being mindful of just how much sugar they're consuming on a daily basis. Now, just going back in terms of kind of a broader level and looking at why genetics is important for nutrition, I would say essential. If we trace the path from virtually any nutritional factor to any health outcome, if there have been enough observational studies, we tend to see this heterogeneity of response, these inconsistencies in the findings. How many times do we read an article that says coffee is good for us, and another day a study will come out and say, no, coffee is bad for us. Another day, coffee doesn't do anything. So there's a number of reasons for these inconsistencies, but an important factor is the genetics of the population that's being studied. So I've tried to illustrate in kind of a black box 3 possible genotypes, where if we can identify whether or not the recommendations we make can improve the health outcome, that's great. In some people, that recommendation might have no effect, and that might not be so bad. But what about in cases where the advice that we're giving actually causes harm? And if there's a way for us to identify those individuals and seek alternate strategies, that's obviously something that we want to consider. So again, you pick your nutritional factor, you pick your health outcome. Sodium and blood pressure. Some people that go on a low sodium diet their blood pressure actually goes up, right? I mean, it goes in the opposite direction. We used to call these people outliers, try to figure out how to get rid of them from our statistical analyses. But more and more, we find that these so-called outliers are actually responding in the opposite direction. And it's not just some statistical anomaly. It's a real biological phenomenon. So if you're one of those outliers and you're responding, the advice that we're giving, population-based advice, is actually causing you harm, wouldn't you want to know that and find out what else you can do to improve your health? So clearly, one size does not fit all. And we've known this for a very long time. In fact, one of the earliest references comes from the 1st century BC, where Lucretius noted that one man's food is another man's poison. What's good for one person might actually be harmful to somebody else. Well, we've come a long way, and now with the modern developments in molecular genetics and genomics, we can actually pinpoint specific regions in the genome that can help us understand why we look and function differently from one another, not only on the outside, but also on the inside. So single nucleotide polymorphisms, or SNPs, SNPs for short, are when we have a single letter substitution. So in the 3 billion base pairs or nucleotides that make up the human genome, where we have a particular letter at one region of the gene, if in someone else they have a different sequence, we call that a SNP, or a common genetic variant. And sometimes these single letter alterations are benign. They don't really do anything. In other cases, they can have profound effects on not only disease risk, but also on our metabolism. And I'll give you a few examples of how these single letter alterations in our genome can have profound effects on how we metabolize specific nutrients and bioactives in our food. Now, before I do that, I just want to make an important distinction between the different kinds of genetic tests, because there's some confusion and I think misunderstandings. I just want to differentiate between the so-called disease risk genes versus what I refer to are modifier genes. So disease risk genes are the ones where if you have a mutation in a particular gene, you are going to develop a disease. So whether that's and these are what we call high penetrance genes. So the gene for Huntington's disease or cystic fibrosis. Those types of genetic variants are fortunately relatively rare, although they have a significant effect on disease risk. And so some of this was popularized a few years ago. If you recall, Angelina Jolie had tested positive for BRCA1 testing. So this is a breast cancer susceptibility gene that has high penetrance. And so she decided to undergo a bilateral prophylactic mastectomy, removal of both breasts, because of this genetic test that said she has basically a 95 to 100% chance of developing breast cancer. Now, when this was popularized, women went out in droves to get tested. And if you test positive, there's something you can do about that, like what Angelina Jolie did. But if you test negative, what does that really mean? What most women didn't realize is the vast majority of breast cancer cases have nothing to do with BRCA1 or BRCA2. Over 90% of breast cancer cases are due to a combination of multiple genetic variants and, more importantly, multiple environmental factors. So if you test negative, that's really a false assurance, because 90% of breast cancer cases will have tested negative for BRCA1. So these are called disease-associated genes. And when it comes to the common forms of heart disease, diabetes, osteoporosis, there is no genetic test that can accurately predict those common forms of disease. There are some cases where they can predict some rare forms of cancer, rare forms of heart disease, like familial hypercholesterolemia, which occurs in a small segment of the population. Where the opportunities are really in the modifier genes. So when it comes to the disease-associated genes, it has been successful for rare diseases like BRCA1-associated breast cancer or Huntington's or Tay-Sachs, but they're not successful for common forms of chronic disease. Again, I think for those, the opportunities are really in the modifier genes. And these are the genes that affect the absorption, the distribution, the cellular uptake, and metabolism of all of the nutrients that we ingest and the environmental factors that we're exposed to. And this is ultimately what can be translated into actionable information, actionable recommendations, something that you can do to mitigate that risk. So when we look to see where and how our genetics can influence our response to diet, as I mentioned, genes influence our sensory perceptions, our likes and dislikes. This determines what kinds of foods we choose to consume or avoid. Genes also influence our appetite. Some people need more of a particular macronutrient to get the same satiating effect. Genes, of course, so those factors influence what we choose to ingest. And once a food is ingested, there's all kinds of genes that influence various metabolic pathways that ultimately determine how much of that nutrient reaches the target cell of interest. So back in 2007, I was giving a talk at the first international conference in Nutrigenomix. And there was a reporter there who, this was in California at UC Davis, and they were working for the Sacramento Bee. The headline the next day was "Genetics May Define Diets of the Future." And they were talking about how this is really a neat field of study, but really futuristic, as though one day we'll be able to determine what we should be consuming based on our genetics. But what's interesting is, at the time, it really wasn't something so futuristic. It was something that we were already doing on a mass scale. And that's by way of newborn screening. So in Ontario and many other jurisdictions, every infant that's born undergoes a battery of tests, what we used to call genetic disorders. But the vast majority of these conditions are actually managed by dietary needs. And so one example is PKU, or phenylketonuria. And this is a condition that results in an inability to metabolize phenylalanine, which is an otherwise innocuous amino acid that's found in a lot of different foods, protein-containing foods. And so the idea that foods are going to be labeled for a person's genetics is again something that we were already doing. In fact, any cola beverage that contains aspartame has this warning label for those with PKU, and it warns them that it contains phenylalanine, because aspartame is a simple dipeptide that half of it is actually phenylalanine. So again, here's a product that's actually labeled for a genetic subpopulation. Now, PKU is relatively rare. Looking at something that's more common, like lactose intolerance, we're seeing a growth in the number of products that are available on the supermarket shelves. I know myself, I have lactose intolerance, but I enjoy consuming dairy. And so we have Lactaid and other types of lactose-free containing dairy products in our house. So fortunately, lactose intolerance is not as severe as some of those inborn errors of metabolism. But in terms of the implications to health of lactose intolerance, we published this study in the Journal of Nutrition just a few years ago. This was with one of my former PhD students, who's also a registered dietitian. And we looked to see what happens in those who have the gene that predicts lactose intolerance. We know they self-regulate and limit how much dairy they consume. And we know that dairy is an excellent source of vitamin D, 25-hydroxy vitamin D. And what you can see here is either in the total population or women and men separately, those who have the white bars, the CC genotype, those are individuals that have lactose intolerance. The heterozygotes that have one copy of each allele are kind of intermediate. Those who have lactose intolerance have a significantly lower amount of circulating vitamin D. And so what this means is that obviously not only are they limiting how much dairy they're consuming, they're also not compensating by getting enough vitamin D from other sources. So obviously it has health implications. This again is the kind of thing that got some media attention. And while it might seem obvious that those who have lactose intolerance might not be getting adequate amounts of vitamin D in their diet, this was the very first study to actually show that this is reflected in their blood levels. And we know that optimal blood levels of vitamin D are important for all kinds of health conditions. So again, the message here is that if you do have lactose intolerance, make sure that you're getting adequate amounts of vitamin D because the evidence suggests that your blood levels are going to be less than someone who does not have lactose intolerance. So shifting gears a little bit to the number one cause of death and disability, not only in the US, but also in Canada, cardiometabolic disease. And this study that was published in JAMA, Journal of the American Medical Association, a couple of years ago, showed that about half of all cardiometabolic deaths are attributed to a poor diet. Now, this was a kind of a general poor diet, like a typical North American diet, high in processed meats and refined sugars, low in fresh fruits, vegetables, and whole grains. But it wasn't really clear what aspect of the diet, and more importantly, in which individuals. So one of the areas that we were interested in terms of heart disease is the link between coffee and cardiovascular disease. Coffee is one of the most widely consumed beverages in the world. It's the biggest source of caffeine, certainly in North America. And the studies that have been conducted have been quite inconsistent. Some studies showing coffee protects against heart disease, others showing no effect, and others showing an increased effect. So what we wanted to see is whether or not we could identify specific genes that can help us predict if coffee is good for us or bad for us. So when you look at a cup of coffee, it's a fairly complex beverage, all kinds of bioactives. Some things like the polyphenols that are antioxidants might actually be beneficial. Other things like the diterpenoids that are known to raise LDL cholesterol might be harmful. So a mixture of some good things and not so good things. So the question we asked is caffeine a component in coffee that might trigger a heart attack? So when we look to see how caffeine is metabolized, it's broken down almost exclusively by the CYP1A2 enzyme, which is expressed in the liver. And it converts caffeine into a more water-soluble substance called paraxanthine. Then that's more rapidly broken down into other water-soluble substances and eliminated from the system. So the CYP1A2 gene, or the enzyme, catalyzes the rate-limiting detoxification or elimination of caffeine. And the nice thing about this gene is that there's a common polymorphism at this position that has a profound effect on enzyme activity or inducibility, as shown on the y-axis. So as you know, we inherit one copy of most genes from our mother and one copy from our father. So if both parents gave us the A allele and made us an AA genotype, that means we have about a 4-fold higher rate of breaking down and getting rid of caffeine. And we can call those individuals fast metabolizers of caffeine. Compare that with someone who has even a single copy of the C allele, which would make them a slow metabolizer. Now, those who are heterozygotes, or those in the middle, might have a slightly intermediate rate of caffeine breakdown, but nevertheless, for most purposes, we can kind of group them together and they say both of them are relatively slow metabolizers compared to a fast metabolizer. So we reasoned that if caffeine is a component in coffee that might trigger a heart attack, then we would expect the slow metabolizers to be at a higher risk. right? Because they're unable to get rid of caffeine as efficiently. If it's going to have any harmful effects, it's going to be in the slow metabolizers where it lingers in the system. Now, initially, we thought that slow metabolizers just consume less coffee or caffeine. But it turns out there was no difference in how much caffeine the fast and slow metabolizers consume. And in hindsight, that's not really surprising, because what you feel when you consume a caffeinated beverage are the stimulating effects of caffeine when it binds to the adenosine receptors in the central nervous system. Okay, you can't just feel how much is floating around in your blood or what your liver is doing. So when we looked in our overall population, this was a big case control study, involved over 4,000 individuals with either an acute myocardial infarction, a non-fatal heart attack, or a control group that was matched based on age, sex, and area of residence. So without taking genetics into account, when we looked at the link between increasing cups of coffee and risk of a heart attack, we found that those who drank 4 or more cups a day had a 36% increased risk of a heart attack. It was statistically significant. This is what we call the multivariate adjusted odds ratio. In other words, we took into account smoking, physical activity, saturated fat, all kinds of confounders, things that heavy coffee drinkers do that could be confounding the link between coffee and heart disease. So even after we adjusted for all those things, we still see this significant effect. Now, the real question we wanted to ask was, is this increased risk due to caffeine? And if it is due to caffeine, we would expect the slow metabolizers to be at a higher risk. So when we stratified our population or separated them between fast and slow metabolizers, that's exactly what we found. On the left-hand side are the fast metabolizers, where you can now see that moderate consumption, 1 or 2 to 3 cups a day, is associated with a significantly lower risk of a heart attack. Whereas the slow metabolizers now, even as little as 2 to 3 cups a day, results in more than a 2-fold increased risk of a heart attack. And the slow metabolizers that drink 4 or more cups a day have almost a 4-fold higher risk of a heart attack. That's about the equivalent risk of a pack-a-day smoker on the risk of heart disease. So we published this in JAMA, Journal of the American Medical Association. This was way back in 2006. And again, it's the kind of thing that, got headlines. This was the front page of the London Times in the UK, why 2 cups of coffee can damage your heart. And the headline on the inside was, gene that could make your next coffee your last. A little bit sensationalist, but again, this is what sells newspapers. What's important really about emphasizing the media coverage is that it shows that the public is being exposed to this kind of science and these kinds of discoveries. So as they meet with their nutrition professional or their healthcare practitioner and they seek nutrition advice and they're given some advice, eventually they're going to be asking, well, how do I know if this is going to work for me based on my genetics? And for a while, we've just been saying, well, overall, the research suggests that it will probably work, but now we should be able to be more specific and say, well, depending on your genetics, here's now what I recommend. So when we published that in 2006, a couple of years later, a group in Italy essentially replicated these findings, but rather than looking at heart attack, they looked at hypertension, which we had proposed as kind of the intermediary pathway. And so even though the cup sizes are a little bit different and the groups are different here, nevertheless, you see the same pattern, right? On the left-hand side, fast metabolizers are protected. On the right-hand side, slow metabolizers, opposite effect. This was also replicated with prediabetes, impaired fasting glucose, showing the same thing, that slow metabolizers have a higher risk with increasing coffee consumption, fast metabolizers, no increased risk. There's been a lot of interest in the role of caffeine as an ergogenic aid for athletic performance, and there's entire books that are written on this topic. And so we decided to look to see if this gene could also determine the effects of caffeine on endurance performance in elite athletes. And so this was work that was done by a former PhD student of mine, Nancy Guest. She's also a registered dietitian. She was the head dietitian for the Vancouver Winter Olympics, and she was also the head dietitian of the Pan Am Games. She actually currently lives in Collingwood, so I think that's about halfway between Toronto and where many of you folks are. And so as part of her PhD thesis, she looked at giving different doses of caffeine to athletes in a randomized controlled manner. And then we published this in the Journal of Medicine and Science and Sport and Exercise. And we showed here that the fast metabolizers actually improved their performance. It took them less time to cycle 10 kilometers with increasing doses of caffeine. Right? So these are milligrams per kilogram body weight. So the lower the bar, the faster they completed their cycling time trial, which means improved performance. So the fast metabolizers actually cycle faster. The slow metabolizers, there's really no difference. But then what we call the ultra slow, those who have the CC genotype, it actually takes them longer, 2.5 minutes longer to complete the 10-kilometer cycling time trial. So again, the complete opposite effects at the highest dose. So 4 milligrams per kilogram, that's roughly 2 to 3 cups of coffee for like a 70-kilogram athlete. So again, kind of thing that got some media attention. Coffee revving up your workout, depends on your genes. That was in the New York Times. And again, in the London Times, a bit of a downer. Caffeine has the wrong effect on the unlucky 10%. So that unlucky 10%, those are the CC genotypes, the ultra slow. The slow metabolizers make up about 40%, and fast metabolizers make about 50%. So again, we're not talking about a trivial proportion of the population. Roughly half the population are fast metabolizers, and the other half are slow metabolizers, with a portion of those being what you could call ultra slow. So shifting gears a little bit and talking about weight management, which is a huge industry, more than a $70 billion industry in the United States alone. And here we're not trying to use genetics to predict obesity, right? Because we don't need that. You can just have a patient stand on a scale, or just to say, look, I need to lose weight. What we're trying to say is, can we use genetics to determine the optimal dietary strategy? Right? There's all kinds of fad diets on the market, right? Low fat, low carb, high protein, keto, paleo, South Beach, you name it. And the reason why these fad diets persist is because they actually work in some people. So the question is, how do we know if that's going to work for me or for one of my patients? Well, oftentimes they do it by trial and error. So if you have a friend, for example, that said, oh, I just went on an Atkins diet and it worked wonders. You're going to expect that it's going to work the same for you, but that's not necessarily the case. So some of the most compelling evidence for the role of genetics comes from this study that was done by Luke Hughes' group at Harvard, where they conducted a 2-year randomized controlled clinical trial comparing the effects of a high protein diet to a low protein diet. So again, something similar to kind of an Atkins diet, maybe a bit of keto and paleo. So here you can see that the effects of a high protein diet on body fat, body composition, so not just body weight, because you don't want to lose muscle mass. So the y-axis shows the percent change in body fat. And you can see that the effects depend on which version of the FTO gene a person has. So those who have either the TT or TA genotype, there's really no difference in how much body fat they lost, whether they were on a low or high protein diet. In fact, if anything, it looks like those on the low protein diet lost a little bit more weight or body fat, but that's not statistically different. But take a look at those on the right-hand side, those with the AA genotype, what we would call the responders. Those individuals who were on a high protein diet lost a considerably greater amount of body fat, and they maintained it 2 years later. This has also been replicated by another clinical trial in Spain that also showed that a high protein diet only benefits those with the AA genotype. And we also replicated this in a cross-sectional study in a multi-ethnic population. And so this was work that was done by a former undergrad student and a medical student, where, again, we weren't able to look at changes in body composition, just cross-sectionally looking at their levels of adiposity. And because this was in a multi-ethnic population, and we had an East Asian cohort where we know that waist circumference is a stronger predictor of cardiometabolic health than BMI, or body mass index. So remember, those with the AA genotype are the ones that benefit from going on a high protein diet. And sure enough, that's what we see here. Those individuals who were not on a high protein diet have about a 10-centimeter greater waist circumference. That's more than a 4-inch greater waist circumference compared to those with the same genotype who were on a higher protein diet. And so the 17% represents the median, so where half the population were consuming less than that and half were consuming higher. And you can see with the other genotypes, there's no difference whatsoever whether you're on a high or low protein diet. So again, only those with the AA genotype can benefit from going on a high protein diet. This was also replicated more recently, just in 2019, in a short-term weight loss trial that was energy restricted but had high protein. And those who had the AA genotype lost a significantly greater amount of body fat in as little as 4 weeks. But again, only those who had the AA genotype. So there are a number of other examples of how our genetics can modify our response to diet. A gene that determines salt-sensitive hypertension, a gene that determines our requirements for vitamin C and folate and B12, and the list goes on. Also, importantly, genes for gluten intolerance and celiac disease. But just in the interest of time, I won't really get into all those other examples. I teach an entire course on this, fourth-year course here at UT. And really just illustrating many more examples. But one question that often comes up is, well, what happens when you give people their genetic information? Will that actually motivate them to do anything about it? Well, we thought that was an interesting question and one that had never been asked before. And on the one hand, a person might say, yeah, I have that gene. It's going to motivate me to eat a healthier diet. But the flip side could be this, this concept of genetic determinism or fatalism, where you think, you know what, it's all in my genetics. There's nothing I can do. Pass me the potato chips and leave me alone. Now, this concept really applies to disease risk genes, because when it comes to the modifier genes, there is something you can do, right? If you're told you have the gene for salt-sensitive hypertension, you need to lower your sodium intake. That's something that you can do. Or if you're told you're a slow metabolizer of caffeine, you need to limit your caffeine intake, that's something that you can do. So we decided to put this to the test, and we did this using the gold standard randomized controlled clinical trial. And we showed that those who receive genetic information compared to the standard recommendation, it actually they understand the recommendations more, they wanted to learn more, and they were more motivated to change their eating habits. So when we followed them up and looked at what happens after 1 year of getting this DNA-based dietary advice, we showed greater improvements in compliance in those who got the DNA-based advice, meaning that those who get genetic information coupled with the right kind of advice actually change their eating habits. And more importantly, they're the ones that need to change it the most. So this was the first study of its kind, showing that our genetics, just knowledge of that genetic information, can change eating behaviors. Since then, a number of other groups have replicated this. And because of that, there was a systematic review that showed that giving actionable genetic information with the genetic report is what leads to the most improved behavior change outcomes. And this same group, that's actually based out of University of Western Ontario, published this recent article in the British Medical Journal, Nutrition, Prevention and Health, where they showed that using the Nutrigenomix test, so our genetic test, on top of the gold standard diabetes prevention program, what's called the group lifestyle balance now, actually shows the greatest improvements, not only in compliance, but also in terms of body fat. They lost a significantly greater amount of body fat, both at the 3-month and 6-month time period. Now, some argue that genetic testing is going to revolutionize the field of nutrition. I think it's going to be more evolutionary. And if you recall, back in 2017, Richard Thaler from Chicago won the Nobel Prize in economics for nudge theory, this idea that you can nudge people to make certain kinds of behavior changes. And this concept really of nudge theory does apply to nutrition as well. And so while genetic testing might not revolutionize nutrition, I think it's really a valuable tool that can help nudge people towards making these healthier behavior changes. And more importantly, these are the ones who would benefit from it the most. So how do we balance the advice that we give to individuals with public health recommendations? Well, there's a growing number of genetic testing companies on the market. Some of you probably heard of 23andMe. Some of these companies are actually no longer in business. I've seen many of these companies come and go over the years. Nutrigenomix, of course, is in the middle, still around. Actually, we just celebrated our 10-year anniversary earlier this year. So where are we today? Well, we need to eat. Some of you are probably having lunch right now. And we currently give dietary advice for healthy eating, but that's based on what I would consider old science, from 15 to 20 years ago. And how much more evidence do we need before we could be providing DNA-based recommendations on a broader scale? So is DNA-based dietary advice ready for prime time? I think if you asked me this 15 or 20 years ago when we first started this area investigation, I would have said no. It's still in its infancy. We want to see the science replicated, but we've come a long way. And I think definitely the answer now is yes. The science is certainly quite robust for some markers. So again, not all genetic tests are created equally. I've seen some tests on the market give wrong information. In fact, in some cases, dangerous information. So there's a little bit of it's a little bit of a wild west in terms of the regulations. So to some extent, it's buyer beware. We and others have shown that the effects are independent of ethnic background. We've shown improved compliance. The information is actionable and personalizable. And again, there's greater consumer awareness and demand for this kind of information, especially as they start reading about a lot of these discoveries that are made, that it depends on your genetics. And lastly, this information, again, has been shown to be more effective if it's delivered through healthcare practitioners rather than direct to consumer or DTC. And because of that, and I'll just give you a quick overview of the company Nutrigenomix, which was established in 2011, but launched in 2012. So again, we had our 10-year anniversary of our launch. It's available only through healthcare practitioners, so not direct to consumer. So a dietitian, a nutritionist, a chiropractor, naturopath, medical doctor, registered nurses, chiropractors, anyone who's a healthcare practitioner who gives nutrition advice can register for an account. And we currently have over 10,000 around the world. Our office is in Toronto, but we also have satellite offices elsewhere. We've supported and continue to support research at various institutions, not just in Canada, but around the world. And our reports are available in several different languages and continue to offer them in other languages as well. So when we launched in 2012, we had just a 7-gene report. We then launched the most comprehensive test for gluten intolerance. And we've actually had some people who were told by 23andMe that you do not have the gene for celiac disease. They did our test, which is more comprehensive, and they had the highest risk. And then they ended up getting antibody testing and biopsy confirmed celiac disease, where 23andMe told them you do not have the gene for celiac disease.
I found this on the web.
Well, that's my Apple Watch talking to me. I don't know why it listens sometimes. In 2016, we launched a 45 gene test, and currently we have our more comprehensive 70 and 77 gene test. We also have reports available in specific formats for athletes. Fertility test, we know that both the diet and genetics of not only the female, but also the male, which influences sperm health, and that influences the likelihood of conception. report for weight management, and also for plant-based eating. So someone who's a vegan or vegetarian, their genetics is not different, but their food options are different. And so if a vegan needs to consume more iron, we don't want to say, okay, have a steak or a piece of chicken. We let them know what the plant-based sources of iron are, so that they can meet those requirements while satisfying their wish to eat plant-based. So healthcare practitioners can go to our website in the top right corner. There's a link to register, so that you can register for an account, get access to our webinars and more training opportunities. We also developed a course with the Dietitians of Canada. It's a 6-hour course. You don't have to be a dietitian to take this course. And so if you're interested, you can just send an email to info@nutrigenomix.com to get a discount code to take this course. We also worked with the American Nutrition Association to develop a course for physicians, nurses, dietitians, nurse practitioners, and other healthcare practitioners if they're interested in getting a certification for nutritional genomics. So just to leave you off then, this article that was in the Wall Street Journal just a few years ago featured not only our research, but also the company, and talked about how this is no longer something that's futuristic. The science is here, the services are here. We need to start moving away from the one-size-fits-all and allowing people to get access to this kind of information to determine what works best for them. So I'll leave you with this quote. Even though the term nutrigenomics is fairly new, the science, again, is quite ancient, and Hippocrates noted over 2,500 years ago that positive health requires a knowledge of man's primary constitution. It's really just an old-fashioned way of saying genotype. And he went on to say that the powers of various foods, both those natural to them and those resulting from human skill. So I think clearly he was referring to personalized nutrition. So I will stop here, and we had given a talk at a conference about a month and a half ago, and we offered them a 50% discount for those who wanted to get tested. And so we wanted to just extend that promo code to anyone here who might be interested. You can just scan this QR code on your phone. We'll circulate the slides afterwards. And this comes with a half-hour consultation with one of our registered dietitians, and this is for the 70-gene report. So I'll stop here. Happy to take questions. I see there are some questions in the chat box that I'll take a look at. In the event that I'm not able to answer all the questions, this is my email at U of T. Feel free to send me an email, and I'll answer questions that way. So again, feel free to unmute your mic and ask me a question. If not, I will go through the chat box and answer questions there.
So I just went through the chat box. There's no specific questions, but if anyone wants to go ahead and unmute themselves, please do so.
All right. I just saw 5 notifications, but I see they're not questions. So.
I just had a question about epigenetics.
Mm-hmm.
I was wondering, when you do your genetic testing, do you have ways to see if the gene is actually active or like on or off? And in your studies, for instance, I myself get a lot of questions about caffeine and stuff like that. Like, were you able to identify anything that would make this gene inducible, or is it always on? Things like that.
Yeah, great question. So again, I have a whole lecture that I teach on epigenetics, and it's really kind of a buzzword that's being bandied about quite a lot, but doesn't really have direct clinical implications. It's really more of a tool that's used in the discovery stage. And the reason is epigenetic changes. So for those who might not be familiar with it, it's something like DNA methylation. So certain regions of the gene can get methylated in the promoter region, and that can either turn on or turn off the activity of that enzyme. And so those kinds of epigenetic changes are tissue specific. So what happens in the liver is not the same as what happens in the muscle or in the adipose or in the small intestine. So an example is something like lactose intolerance, right? So again, I have that gene. Everyone in my family has that gene. And so we're born with it. But why is it that children can generally consume milk? And it's not until they reach, 7, 8, 9, 10 years old that those who have lactose intolerance are no longer able to tolerate. Again, I remember when I was a child, I was able to drink milk. And then when I, got to middle school, I knew that I couldn't drink it anymore. And the reason is because there's an age-dependent epigenetic change that happens in those who have the C allele. So those who have the genotype, the CC genotype, that sequence gets preferentially methylated, and that causes the promoter region to stop working, and the gene for lactase gets shut down.
Right.
And so you don't have to take a biopsy of someone's small intestine to see if it's methylated or if there's an epigenetic change. You just need to measure their genotype and know that if they have that C allele, that will get methylated and it will get silenced. Now, looking at the CYP1A2 gene that affects caffeine metabolism, having the C allele versus the A allele means you have lower activity throughout the course of your life. Now, there might be some instances where expression is higher or expression is lower, but on average, over the course of your life, if you are a slow metabolizer, might not mean that you are slow today or in this instant, because there are things that can increase its activity. And ironically, the number one thing is smoking. Smoking increases CYP1A2 activity. But we're not going to recommend that people smoke so that they can become fast metabolizers of caffeine, right? But there are other things that have the opposite effect, like hormonal contraceptives. They actually compete with the CYP1A2 and decrease enzyme activity. And so that's why women on hormonal contraceptives are generally all slow metabolizers. Doesn't matter what their genes are, they're not able to get, and that's why hormonal contraceptives have been linked with a higher risk of heart disease, not just among smokers, but just in general. And a big part of that could be because a large part of the population is also drinking caffeine. And because the hormonal contraceptives are effectively making everyone a super slow metabolizer, that's causing adverse effects. So there can be some things that kind of turn on and off genes over the course of your lifetime, but you don't really need to concern yourself with that, because you really need to know, what are my genetics? What am I born with and what am I going to die with? And overall, for the course of my life, do I need to worry about caffeine or am I lucky? Do I need to worry about sodium? Or again, am I lucky? Doesn't mean I can eat as much sodium as I want, but that's not something that I need to worry about as much because I'm lucky. My genetics is efficient and it helps me metabolize that particular aspect of the diet.
That's super interesting. Is there anything that you've seen with the, what was it, like GLUT and TSR? One or I don't know, I don't remember exactly, but like for like the glucose genes, did you see that anything influences that as powerfully?
Not as much as in terms of activity, but again, in different populations, we looked at a lean population, we looked at an obese population, we looked at a European population, a multi-ethnic population, and in all of those who have the GLUT2 and TAS1R2 consume more sugar on average compared to someone who doesn't have that genetic variant. So again, the effect size of that gene might be bigger in one ethnic group or bigger in one sex over another, but it doesn't really matter. It's because other things also influence that trait or that outcome. We're not saying that genetics explains it at 100%, but again, compared to not having any genetic information where the advice is just, everything is bell-shaped. Question is, are you at this end of the spectrum or that end of the spectrum? And instead, we're just kind of lumping everyone and treating everyone like an average person when many of us are not.
Right. Well, thank you so much for the presentation. That was very, very interesting for sure.
My pleasure. I see Becky's asking a question here. As a healthcare provider registered with Nutrigenomix, how do I get one of my patients into your testing program? So when you register for an account, you'll get kind of a welcome email with all kinds of information from our practitioners. And then when you want to order a test, we actually offer a free drop shipping which we started during the pandemic because a lot of practitioners closed their physical location. So we can actually ship a kit directly to your client. If you do not have the nutrition expertise to be able to review the report, you can still refer clients, and one of our in-house dietitians will provide that consultation and do that report review for you. So you can just send an email to info@nutrigenomix.com. I believe recently we also started kind of like an affiliates program for some healthcare practitioners who, again, don't feel comfortable to provide dietary recommendations or don't feel as qualified. We would have, but they still want to refer their patients because they recognize the science and they recognize the value and the power of it. They can refer their patients to us, and one of our dietitians will provide the consult. And Ameline, I think you're asking about replay or access to these slides. I believe it's being recorded. This is being recorded. So I think Kathy will let you know how to get access to the recording, and I'll also provide a PDF of my slides afterwards.
So we do actually have a YouTube channel. If anyone wants a link to it, I can send it to you so you can access it directly. Any other questions? I'm wondering what the cost is for patients to access this.
So I showed there on the last slide, again, with a dietitian consult, the price around $399, so just under $400.
Okay, perfect.
And again, the genetics is not going to change, so it's a one-time test. It's not something that needs to be repeated multiple times.
Any other questions? Hearing none. Well, thank you very much for presenting today. That was very interesting. I knew nothing about it, so